Now we're prepared to start talking about the urinary system. And we needed those bits. We've talked about the pressure side of the urinary system, but we hadn't yet talked about the fluid side. Now we have the pieces together so that we can start looking at this system. There's your objectives for this. The kidney does a lot of things, and not all of the things are particularly famous. In some ways, the kidney needs a better PR company to work for it. We all know about the top one, that the kidneys are responsible for excreting metabolic waste products and quote-unquote foreign chemicals. What the heck are foreign chemicals? Medications, right? So medicines, most of the medicines that we give our patients end up leaving the kidney or leaving the body through the kidney. So that's number one. Number two, regulation of water and electrolytes. In other words, how much water is there in the body? That's the kidney's job to control. And what's in that water? In other words, what's the electrolyte concentration? The kidneys have a remarkable ability to selectively control potassium, sodium, calcium, magnesium, phosphate, and so on, excretion and reabsorption. So they're very good at managing electrolytes. The kidneys are responsible for controlling the body's fluid osmolality and osmolarity and electrolyte concentration. Now, they have some help with this from the pituitary gland, but it's ultimately the kidney that has to do the chemistry of controlling this. Arterial blood pressure, as we have talked about through the renin-angiotensin-aldosterone system, is largely under the control of the kidney's endocrine system. Yes, the vasomotor system is playing a role in the seconds to minutes, but over days to weeks, it's the kidney that's controlling arterial blood pressure. Acid-base balance. The kidney and the lungs both participate in this, but the lungs are kind of a one-trick pony. They can only deal with CO2. They can't do anything else. The kidney can deal with any kind of acid-base challenge. So the kidney can create alkali, it can create base, it can create acid, it can create all the different permutations of that. The kidney is responsible for regulating erythrocyte production. That's erythropoietin. And it does so in response to oxygen delivery. When oxygen delivery to the kidney is poor, erythropoietin is stimulated and the body makes more red cells. Secretion, metabolism, and excretion of hormones. We'll talk about hormones at the end of this class, but hormones are the chemical messengers. And in order for a messenger to do its job, it has to be present for only a short amount of time. So our hormones need to be produced, do their job in the body, and then be removed, right? And it's the kidney's job principally to remove those hormones, at least the water-soluble ones. The fat-soluble ones like progesterone, estrogen, testosterone, those tend to be removed by the liver. And then finally, one that nobody ever thinks about is gluconeogenesis. There's a biochem word for you, right? So this is the production of new glucose from something else. That's gluconeogenesis. Remember that we have the ability to produce glucose from protein. We can also produce glucose from fats. Both of those fall into the category of gluconeogenesis. Now, the liver gets most of the credit for this in the body, but the kidney does participate particularly in transforming protein into carbohydrate. And the reason the kidney is doing that is, number one, the kidney has plenty of blood flow and plenty of oxygen for this chemistry. The other bit is when we transform protein into carbohydrate, we end up with nitrogen molecules that we have to get rid of. Well, we get rid of the nitrogen into the urine as urea, and that's taking place in the kidney. So it makes sense to put gluconeogenesis in the kidney because it's the site of the byproduct excretion, right? So numerous things that the kidney does, and it does it with millions and millions of these structures called the nephron. Functional unit. Remember we talked about the functional unit of an organ system is the smallest unit that accomplishes what that organ system does for the body. The nephron is the functional unit of the urinary system, of the kidney. Now, similar to the alveoli and the acinus in the respiratory system that we talked about in MedPath, in the kidney, the nephron, this is not a cell, all right? This is a structure made up of many, many, many cells, okay? It isn't even a tissue. It's technically an organ, right? It's just we have lots and lots of them in the kidney, so we don't really think of it as an organ. But we have multiple tissues working together to accomplish a goal. So the nephron is divided into different sections, both because of its anatomy and because of its physiology, because of its function. Over the next week and a half, we're going to go through each of these sections and what it does. Okay, so remember, this is overview section here. So I'm not going to go into all the detail because otherwise that would be my talk for the next week and a half, right? But we have our renal corpuscle is how it begins. The corpuscle is the glomerulus and the capsule that surrounds it. From there, we enter into the proximal convoluted tubule. That's this part. Once we start to head down and back up, that's the nephron loop, the down and up bit. After the loop, we have another section of convoluted tubule that we call the distal convoluted tubule. Then we enter into what's called the collecting system. This is urine on its way to becoming the final product, right? And as you see with the arrows in this diagram, different things are happening at each of these different spots, okay? So ultimately, we have three processes that are in play here. Filtration, which occurs only at the glomerulus, secretion, and reabsorption. All right, that we have three processes that make all this happen, and we'll unfold those processes as we go here in the next week. So renal phys, ultimately it's about creating urine to get rid of waste products. That's the goal. But what makes it more challenging is we want to control the levels of fluid and electrolytes while we do that. And we want to hold on to molecules of value. One of the most important things that the kidney does is not get rid of stuff. All right? The way the kidney works, everything smaller than a protein is filtered. In other words, it's pushed into the nephron. Well, there's a lot of stuff smaller than a protein in the plasma that we would hate to lose. Things like glucose and protein, or not protein, but amino acid, electrolytes. These are things that get pushed into what would become the urine, but the body reclaims those things because they're useful. So we can see a little bit of how the kidney works by looking at the in and out of it. All right, so if we look at this table, plasma is what goes into the nephron. Okay, what comes out of the kidney, what comes out of the nephron, is urine. These two things have a number of important relationships to each other. Okay, so let's look at sodium, for example. Sodium in the plasma, 135 to 145. I've been talking about that already today. Well, in the urine, the sodium in the urine is hugely variable, from 40 up to 220. This is because our dietary intake of sodium varies, right? You know, you can have the bathtub of popcorn, right, on a day, and you're going to excrete a lot more sodium that day. Or you can be on an ultra-low sodium diet and have almost no sodium intake, and then your urine sodium will be very, very small, right? Because the body is controlling that. So it's an example of variable excretion. The body will hold on to a thing that it needs, and it will dump a thing that it doesn't need, right? So potassium falls into that category too. Very narrow range in the plasma, huge range in the urine. Remember always when looking at potassium that the vast majority of potassium in the body is inside of cells. You're never going to see it in the plasma, right? So our intake of potassium has to be balanced by an output of potassium in the urine. So that's going to vary. If we increase our potassium intake, more potassium will enter the urine. If we decrease it, we'll have less. Chloride is about the same, but it's going to follow sodium for the most part. But now look at bicarbonate. Bicarbonate is a particularly valuable molecule to the body because it's part of our acid-base balance. It's our primary extracellular buffer against pH change. So the kidney doesn't want to get rid of bicarbonate. So we see that bicarb is in much less amounts in the urine than it is in the plasma. Now, I would say this can, in fact, vary. You know, earlier today we were talking about metabolic compensation for respiratory acidosis or alkalosis. That's one of the variables that can change. The kidney can excrete more bicarbonate or less bicarbonate according to what the body's pH status is at any given time. All right, if we look at the metabolites, we see a pattern. There's lots of them in the plasma, because plasma is doing its job delivering nutrients to the cells, right? But there's almost none in the urine. Why? All these things are valuable to the body, so they're all reabsorbed. Even though they enter the urine, they don't come out of the body. They are reabsorbed back into the blood for use by the body. So like glucose, for example, even though there's a fair bit of it in the blood, in the healthy person, there's almost zero in the urine. Okay, so that's powerful reabsorption. Waste products is opposite of the nutrients. Here, we have relatively low concentrations in the plasma because remember, one of the jobs of the cardiovascular system is to wash away waste products, right? And that's all powered by diffusion. So we have to keep the concentration of waste products low in order for diffusion to drive the wastes into the blood. Now, when we get to the kidney, though, we want to have lots of waste product in the urine because that's how we get it out of the body. So like urea, for example, is the primary way we get rid of nitrogen. You know, remember, our bodies have very poor systems for dealing with nitrogen, okay? So about all we can do with it as mammals is put it in the urine as urea. Now other organisms metabolize nitrogen just fine, but we don't. So we see lots of urea in the urine, lots of creatinine, fair bit of ammonia, fair bit of uric acid. Those are all nitrogenous waste products, ways that we get rid of nitrogen out of our metabolism. All right, we'll talk about the three processes and then we'll call it for today. All right, so filtration, secretion, and reabsorption. Filtration, we've kind of already talked about. I said the kidney works like a pressure filter, you know, like a Brita water pitcher does, where you put water in under pressure and the stuff comes out. Well, that's filtration. We put blood pressure across a membrane. That membrane has tiny little holes. Well, this blood pressure, together with the tiny little holes, solutes are going to be pushed out. Anything smaller than a protein will get pushed out. Now, of course, that includes water too, right? Water's smaller than a protein, so it'll get pushed out as well. Filtration only occurs in one place, and that's at the glomerular membrane, in the corpuscle. And it's filtration that begins the formation of what will become urine. All right, so at the beginning we call it filtrate because it isn't quite urine yet. Secretion is the process of moving molecules from the blood into the urine. We're going to see this in our waste products, right? We're going to secrete a bunch of urea. And when needed, we're going to secrete electrolytes. You know, so let's say you have a very high calcium diet. Well, you're going to secrete extra calcium in order to keep that calcium level where it belongs. And then finally, reabsorption is what we do with our filtered nutrient molecules, like glucose is the greatest example here. Glucose is freely filtered. It's pushed out because it's smaller than a protein, but it is readily reabsorbed. That glucose is moved from the filtrate back into the plasma so that it can be used by the body somewhere else. All right, so these three processes, we're going to see filtration plays a role in the corpuscle only. Secretion and reabsorption happen through the rest of the nephron. All right, and we'll talk about how that works. At this point, it'll be next week. We have the amazing nephron, right, as I like to refer to it. And we talked last time about the three processes: filtration, reabsorption, and secretion. Literally, everything that happens in the kidney can be described in those three terms, right? At least in terms of urine production. So what I have here is basically those three processes shown as arrows across the anatomy of the nephron. So you'll notice the green arrow, filtration. Filtration only occurs at one place, okay? Because the only way we can have filtration is we need to have blood, right? Remember, blood is the source of the filtrate. What comes out of blood we call filtrate. What comes out of the kidney we call urine. All right, so the process of urine production goes blood, filtrate, urine. The reason we don't call it urine when it enters the glomerulus is it's not urine-like at all, right? It has a lot of glucose in it, a ton of water, like it just doesn't match. So we're going to call the fluid moving through this system filtrate until we get to about here, right? Now, technically, urine is what comes out of the kidney, but in the collecting system, we often do still refer to it as urine, okay? Just to be specific on the details. But here at the glomerulus, we have blood pressure going in. We have a filtration membrane, which we're going to talk about later today, that pushes everything smaller than a protein, gets pushed out into the glomerular capsule. That's what I mean by filtration. Small molecules pushed out under pressure, okay? Now from there, the rest of the modification of this fluid will take place using the other two processes, reabsorption and secretion. In reabsorption, those are the outbound arrows, right? So that's material that's being removed from the filtrate and brought back into the blood. Typically, these are things the body finds valuable, right? So your glucose, your electrolytes, these are things that are hard to find in the environment, so we don't want to waste them in the urine. So those are the reabsorption parts. And the inbound arrows, okay, that is going to be secretion. And in secretion, what we're doing is we're taking molecules from the blood and we're putting them into the urine. Okay, so that we're adding materials to the urine. So this is typically electrolytes or waste products, right? Now, why electrolytes? Remember, sometimes we have excess electrolytes that we have to get rid of. Like if we have a high sodium diet, for example, we have to get rid of that extra salt. So that becomes secretion. From your book, we have this kind of functional diagram of that same nephron, sort of just made simpler. So we have our glomerulus. Now, the glomerulus has an interesting vascular pattern. It has two arterioles. So far we've looked at artery goes to arterial, goes to capillary, goes to venule, goes to vein, right? Well, here we enter our first portal system, okay? A portal system is a system of vessels where there are two capillary beds between heart and back to the heart, okay? So think about it. In a normal system, there's one capillary bed. Well, in a portal system, there's a capillary bed, and then later on, there's another capillary bed. All right, so where are the two in this case? Blood comes from the heart through the afferent arteriole, through the glomerular capillaries. These are capillaries, right? So this is our first capillary bed. Then blood comes back together into another arteriole, okay, not a venule, but another arteriole. And then from there, it goes back out of the kidney, and then we get a second capillary bed that surrounds the nephron that we call the peritubular capillaries. Then we get into venules and veins. We're going to see two other examples of portal systems this semester. One is in the GI tract, that's the hepatic portal system. The third one we'll see in the endocrine system when we talk about the pituitary's portal system. All right, so we got three of these. What makes the kidney portal system unique is we have two sets of arterioles. Normally, even in a portal system, we don't have two sets of arterioles. But this is an important anatomic reason for that, and that is each of these arterioles can control its own vasoconstriction. So I like to think of these afferent arterioles and efferent arterioles are the valves of this pressure filter right here, okay? So if you think about, like a garden hose, right? You've got a valve at the house, and let's say you put another valve, and then a valve at the house, hose, sprinkler, then you put another valve at the end, right? Well, if you open the valve at the end and the valve at the house, how high does the sprinkler go? Come on, put it together. Very little, right? Because it's all going out into the ground. Well, that's the efferent arteriole, is the one at the end. Now let's tighten down the efferent arteriole, leave the one at the house on. Now we've got great big sprinkler action, right? It's because we've increased our pressure in the arteriole. That's a little bit how the body controls this system. There's two valves. You can let pressure out the end, or you can reduce pressure going in the first place, okay? So we'll talk about controlling glomerular filtration another day. But we have afferent arteriole and efferent arteriole. You all know I hate these words, right? Afferent and efferent, I wish they'd have just called them in and out or something. Okay, but afferent goes in, efferent goes out. The way I remember this, efferent makes effects. In other words, effects are something coming out of a thing, right? So don't know if that helps. It somehow helps me. But afferent and efferent, glomerulus in the middle. Then we come back to the peritubular capillaries and so on. All right, so number one is our filtration, pushing fluid out at the glomerulus. And then we have reabsorption and secretion. Remember that reabsorption and secretion, the other end of those processes are the peritubular capillaries, blood. So when we talk this week about reabsorption and secretion, it's either going back into the blood or coming out of the blood. That's the source of reabsorption and secretion. And then, of course, the final excretion is out through the ureters, bladder, and urethra, right, and out into the world. And we'll talk about that at the end. Okay, so if we do our structure diagrams for the three processes, you can kind of see what's happening. So in A here, we have a substance in the blood that is only filtered. Okay, so it passes through the glomerulus, it's pushed out because it's a small molecule, and then it doesn't move after that. An example of a molecule like this is creatinine. Okay, creatinine is filtered but not reabsorbed or secreted. It's one of the reasons we use creatinine in our evaluation of kidney function, okay? Because the only thing that affects creatinine levels is glomerular filtration, the first part of the urine production, okay? So we use it for that. Not a lot of substances fall into that category, though. Creatinine is a bit unusual. Another thing that you'll see do this is medications. A lot of times are filtered but are not reabsorbed or secreted. All right, so then we have an example where we have partial reabsorption. So a molecule is pushed out, some of it is reabsorbed, some of it is left in the urine. This would be an example of sodium, let's say, right? So sodium is freely filtered. It's pushed out because it's a very small molecule, but depending on the body's needs, it's going to reabsorb at least some of it, okay? Here we have filtration with complete reabsorption. So a molecule is pushed out by filtration, but then through active reabsorption, it is all reabsorbed so that it does not appear in the urine. A good example here would be glucose. Glucose is freely filtered, but it is completely reabsorbed in the normal case. Now, if your blood glucose is greater than 300, you'll start seeing glucose appear here because of you overwhelm the receptors. That's saturation, you overcome saturability. And then here we have a filtration and secretion. This is what we see in molecules that don't filter particularly well, all right? So in substance D, we've got a small amount of that molecule is filtered, but then later on, much more of it is secreted into the urine. So an example here would be maybe an electrolyte that we have too much of. So let's say our calcium status is we've taken in a lot of calcium. Well, we're going to filter some calcium, but the body can secrete much, much more if need be, okay? So our three, just examples of the three processes. Okay, so there is some anatomy that matters in the kidney. We essentially have two regions. We have a cortex region and a medulla region. And if you, oh, come on, where's my drawer? Draw one of these and, nope. Well, the pen stopped working, folks. So you're going to have me drawn on the board. All right. Kidney, right? This is the cortex, is the outside cortex. And this is the medulla. Now, these are not terms that are specific to the kidney, all right? Cortex just means outside, medulla just means inside, because like when we talk about the adrenal gland, we're going to see the same terms, all right? But when we look at the kidney, to take this picture and now zoom way in, this is the cortex, this is the outer part, this is the medulla, this is the inner part, okay? We have two kinds of nephrons. This is a nephron right here, and this big thing is a nephron right here. Did that just go off?
Yeah.
Just randomly?
Yeah.
Okay, good. All right, notice the cortical nephrons are quite a bit smaller than what we call the juxtamedullary. So the big word, juxtamedullary, they're for big nephrons, all right? Juxta means near to, so juxtamedullary, all that's saying is nephrons that are close to the medulla. That's what it's describing. The nephrons that are close to the medulla are much longer. They have a much longer loop of Henle, or nephron loop, as we prefer to call it now. And what that means is that long loop makes it much easier to absorb additional water. So we're going to talk at length about water reabsorption later. But for now, know there's two kinds of nephrons: shorter cortical nephrons, longer juxtamedullary nephrons. There are oodles more of the cortical nephrons than there are of the juxtamedullary. It's like 80%, 20%. Some will say 90-10. But that's okay, it still works, as we'll talk about later. The other thing I want to point out in the location matters slide, all right, so you have your cortex, then you have the medulla. In the medulla, okay, you have a osmolar gradient, which is going to develop. So down here in the inner zone, the interstitial space here has a very high osmolarity. In the outer zone, it has a high osmolarity, but less high than the inner zone. And in the cortex, we have normal osmolarity. Now, I'm setting something up for later, all right? We're going to talk about how as urine passes down into the concentrated area and then back up, that helps us to be able to conserve water. And I'll explain the mechanism behind that later. But for now, just know lots of osmolarity, medium osmolarity, normal osmolarity, okay? And then if we add the capillaries to this picture, so this is a cortical nephron, right? This is a juxtamedullary nephron. If you look, I want you to zoom in and look at the patterns of the capillaries that surround the nephron. Do you see how here they're kind of going every which way? But here in the juxtamedullary, the capillaries tend to go down and back up. They follow the nephron loop. You don't have connections across here. See that? That's actually really important. We call these capillaries the vasa recta, and they only exist in the juxtamedullary nephrons, okay? And the vasa recta, they evolved the way they did because of the concentration gradient in the kidney. Down here, the area surrounding this nephron and capillaries right here is very highly concentrated, very high osmolarity. If blood from this area were to just go right back into the circulation, it would cause massive shifts in osmolarity. So in the juxtamedullary nephrons, the blood has to go back up the concentration gradient back to the cortex before it empties into the rest of the circulation. Because while it's going back up, its osmolarity is going to correct and come back to normal, all right? We'll pick up more on this when we get to our urine concentration slide, but this is sort of the only time I talk about anatomy, so I wanted to introduce that now. All right, this is kind of a preview table, all right, or a summary table. I'm not going to go through all this right now. This will be one of those once we finish this unit on the kidney, this will be a table to come back to review the different parts, okay? Because literally we're going to talk like a lecture or a half a lecture on each of these as we go. So this is kind of the compact version. All right, let's look at the renal corpuscle. The renal corpuscle a corpuscle just means ball-shaped, right? So this is the ball shape at the beginning of the nephron. It's oftentimes referred to as the glomerulus, but that's technically not correct. The glomerulus is this bit inside. The corpuscle is the capsule and the glomerulus inside. Okay, just to you know how anatomists are. You know, you've got very specific words, okay? So this is our glomerular capillaries, afferent arterial, so blood coming from the heart, going through here. Here's our first valve, right? And then the capillaries in the glomerulus have formed a kind of tuft or ball or round shape, for lack of a better term, that has blood passing through it, okay? And then the blood comes back out here. Now, surrounding these capillaries are cells called podocytes. That's these little purple cells right here. And the podocytes have little feet processes called pedicels. The pedicels of the podocytes kind of wrap around each other like interlocked fingers. And they make a filtration membrane. They make a barrier between blood and outside that's too small for everything but a protein to pass through. Okay, so this kind of knot is where blood's going to pass through and fluid will get squeezed out in between these little slits called the filtration slits. So if we zoom in and look at that membrane, okay, so we're going to take a piece of the glomerulus and we're going to look real close at it. So here, this is your capillary, right? So what we've got here is blood flowing through there. Now remember the scale. Capillaries are so small that red cells have to squeeze through. Okay, so we're talking very small tubes here, right? But each tube is a complete tube. Now, making the wall of that capillary is this bit right here. This is the capillary endothelium. Now, in the kidney, the capillary endothelium has pores. It has holes in it. So it isn't a complete tube. So for a typical capillary, you can imagine like a garden hose, right? For a kidney capillary, it's a garden hose that's had a bunch of holes drilled in it. All right, so you have literally holes, fenestrations we call those, of the glomerular capillary. So blood has to pass through these fenestrations, through these holes, in order to get out. Materials have to pass out. Now, the next layer underneath the capillary endothelial wall is what's called the dense layer. The dense layer is essentially the basement membrane. It's the fused basement membrane of both the endothelial cells and the podocytes. And if you remember what the basement membrane is, it's basically a proteinaceous membrane, right? Now, it is continuous, but it's not particularly resistant to small molecules passing through. So even though the basement membrane, while it doesn't have holes, it's more like a meshwork than it is like a solid wall, okay? So materials can pass, small materials, things smaller than a protein can pass through the dense layer. And then the last layer of filtration is the pedicels of the podocytes. Okay, so each one of these little triangles in my drawing is actually one of these little feet processes of the podocytes, right? Do you see how they kind of wrap like fingers next to each other? In between the pedicels of the podocytes, we have these little spaces. Those are called filtration slits. So our membrane is made of 3 layers. The endothelial membrane, which is fenestrated, the dense layer, which is the fused basement membrane of both, and then the pedicels of the podocytes on the outside. All 3 work together to do the same thing, and that's to resist the motion of large molecules, okay? Quite frankly, it works just like the filter paper you've used in past chemistry classes, okay? Filter paper allows water, salt, many things through, but if you try to put through protein, it gets stuck in the filter paper, right? Same kind of thing. The glomerular filtration membrane evolved to restrict the flow of large molecules. So what enters the capsule, remember the capsule's out here, is going to be everything smaller than a protein. Is a red blood cell smaller than a protein? No, thank you very much. Right? A red blood cell is made of lots and lots of proteins. It's a behemoth compared to a protein, right? It's a great big huge thing. Now, hemoglobin, much, much smaller than a red cell, right? Is it bigger than a protein? Yeah, because it is a protein, right? And it's a big protein. So hemoglobin, for example, cannot make it through this filtration membrane, right? Only small things. Water, is water smaller than a protein? Absolutely it is. Is sodium, calcium, do you see where I'm going? Glucose, right. Almost everything else is, but if it's bigger than a protein, it never enters into the blood, or it never enters into the capsular space in the first place, at least in normal kidneys, okay? Remember, we have diseases that affect just about everything. We have a disease that we have many diseases, that, and Kara's going to teach you about them in MedPath later on. We have many diseases that affect the glomerular filtration membrane. One of those is what we call nephrotic syndrome. In nephrotic syndrome, this membrane breaks down so that proteins can cross. So then all the proteins from your plasma end up in your urine. And you can imagine what that does to your plasma oncotic pressure, right? Okay. There. Okay, so I do like to take this time just to talk to you a little bit about dialysis, because I don't think you learn about it any other time in here. So this isn't testable material. You can say, don't need to learn this, just listen for a second. But I wanted to talk to you a little bit about how dialysis works, right? Okay, so you now know everything about the kidney, right? Not quite, but you know generally how it works. We have a pressure filter, and then we have reabsorption and secretion. Okay, so how the heck do we make a fake kidney for a patient whose kidneys have failed? Well, what we end up doing is, first we're going to need to have a lot of blood, just like our actual kidney. Remember, kidneys get like 20% of the cardiac output because not only are they oxygen hungry, but they also have a job to do with the blood. They have to filter the blood. All right, so in a patient on dialysis, first we have to get a lot of blood out of them. Well, that's not particularly easy, so we cheat, and we make what's called a shunt. We find their radial artery, and we put a plastic, smooth shunt in that connects their radial artery with their, what is it, ulnar vein? You guys probably know better than I do, one of those veins in here, right? Okay, so what we get is we get blood that very rapidly goes from arterial side to venous side. Very little resistance here, right? So blood flow is going to be very high. You know that from the physics we've been talking about. So lots of blood flow through the shunt. So high, in fact, that you can hear it move. So if you have a patient who's on dialysis that you're encountering next year, ask them where their shunt is. They'll know, believe me, they'll know. And you can listen over the shunt, and you'll actually hear the blood moving through the thing, at least in a healthy shunt. Okay, one of the restrictions on dialysis is eventually the shunts clot off. And now you got to go find another one somewhere else, and eventually you run out of vessels. Do you see the problem? Okay, so first we get an access point. So now that we have this big burly amount of blood moving through this tube, we stick a needle in one end and a needle in the other. Okay, so now we're going to take blood out of the patient. Okay, we're going to run it through a pump. We're going to put it through what's called a dialysis chamber. More on that in a minute. Then we're going to return it to the patient. Okay, so we're not taking a lot of blood out. We're just taking blood out and putting it back in. Now, what do we do with the blood? How do we actually make a kidney? Well, we run the blood through what's called a semi-permeable membrane. So this isn't just a normal plastic tube that this blood is running through. It's a very thin tube that allows the passage of molecules smaller than a protein. Oh, look at that. Does that sound familiar at all? Because it should. All right, so that's a dialysis membrane. We run the blood through a dialysis membrane. That alone wouldn't do us any good, right? It would still end up back in the patient. So then we take that dialysis membrane with the blood inside, and we put it in a fluid. Well, in that fluid then, we tweak the amount of sodium, potassium, calcium. We tweak the electrolytes, and we tweak the waste products, so that as blood passes through this solution surrounding it, it exchanges with it, right? So for example, you're experts on diffusion now. If the patient's sodium level was too high, okay, we would put in the dialysis fluid a sodium concentration that was lower than the patient's, so that sodium would flow out of the blood and into the solution, thus lowering the patient's sodium level. See that? For potassium, we might have almost no potassium in that solution, so that as blood passes through the dialysis chamber, potassium is removed from the patient. Now, of course, you have your waste products, which is primarily what this is for. In our dialysis fluid in that chamber, we're not going to have any urea, right? So as blood passes through that, urea is going to end up in the fluid until it equilibrates, at least on the way out. And then, that fluid is going to keep getting full of stuff we're taking out of the patient, right? So we circulate it. We drain this fluid out and we put new fluid in as the blood is flowing through it. And we essentially are moving molecules into and out of the patient using diffusion, using the concentration gradient. So it's a fairly elegant system. Now, the downside with it is it takes a long time. Remember, your kidneys are working all the time. A patient with advanced renal failure, they could end up on dialysis 3 to 5 times a week, 3 to 4 hours a day. So you can imagine that's pretty much like a life, right? So dialysis, while it's functional, it is a huge barrier to people's quality of life. Now, you might also wonder, how do we do this in kids? Well, we can do this same process with kids. One of the things that's a little more concerning with kids is at any given time, you've got a fair amount of blood that's outside the body, right? So both in adults, but definitely in children, we oftentimes have to prime the dialysis machine with blood before we hook up the patient. So essentially, they're getting a partial blood transfusion, and the risks that are associated with that. Because otherwise, if that's, let's see, 250 cc's of blood, well, that's a quarter of a baby's blood volume, right? So you can't really do that. Now, another way that we can do this, and I have 5 minutes to talk about it, is we have a place in the body where we have lots of blood passing through a semi-permeable membrane. Anybody know where that is? Yeah, you're on the right track. The GI tract, right? In the GI tract, you have lots and lots of blood passing through the walls of the small and large intestine, right? The small and large intestine are thin-walled tubes. Well, wouldn't it be great if we could use that blood passing through that semi-permeable membrane to do our dialysis? And we do, particularly in children. We call it peritoneal dialysis. And here, instead of taking blood out of the patient and running it through a dialysis machine, we take the dialysis fluid and we put it in the patient's abdomen, right? So we mix our dialysis fluid, we put it in there, we let it sit overnight, blood passes through this fluid, exchanges its molecules, right? So the urea ends up in the dialysis fluid in the gut, and like sodium enters and all that. Then in the morning, we pull out that fluid, and along with it comes all the waste products. Now, I'm oversimplifying. We actually exchange the fluid several times each night, right? But then when the kid wakes up in the morning, their urea is down, their potassium is down, their sodium is stabilized, and their fluids are stabilized, right? So it means that we don't have to take blood out of the patient, and it's much better tolerated. Now, why don't we use that in adults? Because the bigger you get, the less gut surface area you have for your body surface area. So peritoneal dialysis, which is what we call the internal version, stops working when you get too big, right? So you either have to have partially functioning kidneys for it to work, or you have to be a kid who's small enough. So that's just a little bit about how dialysis works. We fake the glomerular membrane with a semi-permeable plastic membrane, and then we tweak the fluid around that membrane in order to get the molecules to move the way we want. Okay, let's do our Kahoot here, and then all right, off we go. I forget how many questions we have in this one. 3. Okay, good. Resorption of useful molecules mostly occurs where? Let's see. I don't know, I didn't say it in class, but we'll spend a whole session on it tomorrow. Yeah, that's proximal convoluted tubules. So the first thing the kidney does is reabsorb all the stuff that is useful to use, and we'll talk about those processes here as we go this week. Loops of Henle are found mostly in what area? The medulla, very good. It's the loops of Henle and their metabolism are what create that osmolar gradient in the medulla. Okay, and that's one of the more challenging concepts that we have to cover this week. Ooh, zany dragon holding on to the lead. A molecule passes through the glomerular membrane and is then moved back into the blood. Which is that? I'm hearing a lot of grumbles. A lot of grumbles. All right, so if it passes through the glomerular membrane, that's filtration, okay? If it is put in the blood after the glomerulus, that's secretion. If it's moved from filtrate back into blood, that's reabsorption. So this is a molecule that is both filtered and reabsorbed, like glucose might be, okay? All right, let's see who our winner is. The purple condor with the 2763.